taking over, so we'll see how that goes. But we're going to be covering clustering and dimensionality reduction. So I think just as a reminder, I am an immunologist that has been experimenting and delving into bioinformatics and computing. So today, what I want to focus on is just some of my own experience with a lot of this and my opinion on what I've gathered from using these kind of techniques that are available to us. So for today, I mainly want to focus a lot more, I guess, on the theoretical side of these things. So what the difference is between both of these, why and how we use them, and then going into some of the assumptions and limitations for them. And when we consider this, I think it's important to appreciate the fact that how we as humans see cytometry data is different to how computers see it. And I think this is the crux of the issue for a lot of us, where we find that the computational algorithms may give us different results to what we ourselves would find when we do this manually. So I think when it comes right down to it with cytometry, it really is just a glorified game of spotting the difference. So, and I think this is probably true for the majority of science as well, that with cytometry, we're really just comparing, different subsets of cells between groups. So this may be between healthy and diseased patients, but we're also trying to differentiate between individual cells to try and identify different subsets. And this is a lot easier to do when we're viewing more dimensions at a single time. So this is just an example here. We're looking at CD5 expression. These are just some PBMCs. This is some mass cytometry data, which is why there's a lot of cells banked up at 0. But what I hope you can appreciate here is that when we look at just one dimension like this, it can be difficult for us to determine where we want to cut off the positive from the negative cells. But generally, to help us out with this, we look at 2 dimensions at once. So here we now have CD3. So what I hope is quite apparent is that we can start to more clearly differentiate between some of these cell subsets. So when we look at more dimensions at once, we can really improve the clarity for differentiating between various cell subsets. And this is obviously a huge limitation for humans. At most, we can look at 3 dimensions, but even then, the majority of the time, we only look at 2 dimensions. And that's mainly because the technology isn't very practical for us to look at data in 3 dimensions. It's quite difficult to do. And the fact is, 2-dimensional plots have been quite useful for decades now. But what I hope you can appreciate, though, is even by going up from 1 to 2 dimensions, we can get a lot more clarity here. And this only increases the more number of dimensions that you can look at a single time. And this is one of the big things that computers are able to do. In theory, they could look at an infinite number of dimensions. And with this, it is really able to provide a lot of power to help differentiate between different cells. This does come with a big catch, though, and when it comes to spotting the difference, computers themselves, they are by far superior compared to humans, but they're too good. And this is just because they're a lot more sensitive to differences within data. And this is why humans are a lot better at pattern recognition. It's just because we're not as sensitive as computers. And hopefully this analogy will help clarify what I mean by this. So if we're looking at 5 different pairs of genes and we want to differentiate between them based on their colors, for humans, it's really easy for us to just divide them into 2 groups, so blue and black genes. If we ask the computer to do it, it can be a lot more specific. So it may split them into light and dark colors. So what I've just got here are different color codes, and that represent the colors of these specific genes. And to a computer, it's able to add a code like this, and then it can directly compare between them. And it will say, this code is different to this code, so these must be 2 different groups of genes. And again, humans can do this, but we generally are lazy. No one really wants to differentiate to this level. And this is where, again, I think humans are better at pattern recognition because we're less sensitive, or we don't pay as much attention as computers do. And this is why I think we get so many discrepancies between what computers find and humans find. And when we look at cells themselves, so this is just some mass cytometry data. Here, each row is a different cell, each column is a different marker. So every individual cell will have a single signal intensity for every single marker present. And again, what a computer will do is it will look at each of these rows and try and find if any of them are identical. And to a computer, if no cells are identical, by definition, they must all be different. And again, I think this is the crux of the issue, where computers themselves are a lot more sensitive to humans when trying to look for differences. And as I said, I think this is why we get so much discrepancy between the two. Another thing that I think is worthwhile to point out is the term whether computers are unbiased or not. And it is true, they are unbiased, but only to a certain extent. And I think it's important to appreciate that not all bias is bad, especially in cytometry. Understanding the biology is really important. And Tom spoke about this yesterday and the day before in terms of when we gate on our cells. In the majority of cases, we're only looking at live cells, we're ignoring the dead cells. That itself is introducing bias to the system. We are actively choosing to only run the live cells through these computational algorithms. But this, to me, is a case where I think that is an acceptable introduction of bias to these systems. And there's multiple other cases like this, but I think it is just being aware that almost every decision we make will be introducing some bias of some of the script. And this is where it's just more important to be aware of how that may be influencing, in particular, the outputting results of these computational algorithms. And the other thing here is that no single approach will ever be able to find all the differences that we're looking for. And I guess this is where we recommend that we use these kind of approaches to complement our manual gating or any of your other conventional analytical methods. By no means is one, I think, better than the other, but if you really want to make the most of it, you use them both together to complement each other. So now we'll move on to clustering and dimensionality reduction. So I think at the core, like in layman's terms, they're quite similar in the sense that they're just trying to differentiate between cells based on their phenotype, but it's generally the outputting results are going to be very different. And as well as this, because again, these are both different methods of trying to find a similar meaning in our results. And again, that's just trying to differentiate between the cells. And this is where these kind of algorithms, as I said, computers are a lot better at spotting the difference than humans. But I think that the computers, when we write algorithms like this, we're trying to make computers think more like humans. And that's why there's so many different approaches that you can do this. Even just between individual clustering algorithms and dimensionality reduction. And we'll get to some different examples later on in this talk. So in this case here, this is just the same data I showed earlier. So each row is a separate cell, each column is a different marker. Both of these algorithms, all they're doing are adding a few extra parameters to the individual cells. So clustering, for example, this is just clustering done with flow sum. It will create a meta cluster as well as a cluster. So each individual cell will be put into different groups. And that's just indicative by this number. At the same time, dimensionality reduction, we have an example of both UMAP and TISNI. All these are just coordinates on the dimensionality reduction plot. So it's quite, I think, refreshing to me when I realize that the data itself, it's very straightforward when you look at the end result, because all it's done is just added a few extra parameters to each individual cell. And this is why we can very easily analyze these results in our conventional cytometry analysis software like FlowGel. At the same time, these are very different. And the key difference to understand between these two is that clustering will group cells together based on their phenotype. And you lose the individuality, but you do get immediate quantifiable results that you can compare between groups. So this is just an example of a spade plot. On this plot, each dot represents a cluster of cells. So each dot has a number of cells within it. And so what you can do is, if you had these plots for two different groups, you can directly compare the level or the number of cells within that cluster, and then you can immediately compare between groups to see whether it changes or not. At the same time, what this figure nicely highlights as well is that these individual clusters can start to be grouped together at a higher level and looking at major cell subsets. So you can still get these nice visualization of the major cell subsets in your data. Now, dimensionality reduction, on the other hand, keeps each cell independent of each other. So you don't get the immediate quantifiable results that you do compared to clustering, but these are very useful for just visualizing the heterogeneity of your data. So this here is an example of a Tisney plot. What I think is really cool here is that on this plot, each dot is not a single cell, each dot is a single star. And what they've done is, this is part of the GLASS study, they've had a look at the elemental components of stars as parameters to try and see whether stars with similar elements will be grouped together. And what they're trying to do is create almost a family tree of stars. So I think it's really cool that these are the kind of things that you can do, not just at the cellular level, but you can do it at the galactic level as well, which I think is really cool. And I think that's another point here as well, is that the idea of clustering and dimensionality reduction, it's not new. These have been around for decades. It's just new to cytometrists in general. And that's just because we haven't really needed them, because we haven't had as many parameters or as many cells as we're starting to build upon now. So why do we use them? Again, the clustering, like I said, it's really useful for quickly dividing up our data into immediate quantifiable subsets, so we can directly compare between cells. Obviously, it's really appealing because you don't need manual gating for the clustering. It is still essential though, and we'll get to that in a bit, but it is very useful for quickly giving you quantifiable results. Dimensionality reduction, on the other hand, as we saw, it can very nicely visualize the heterogeneity of your data. And this is useful just to get an idea, I guess, of all the major cell subsets that may be present within your sample. And you can also compare this between groups to get an idea of whether their levels are changing or not. Both of these are obviously a lot more objective than manual gating. The catch behind this, of course, though, is that they are a lot more susceptible to variability. And as we saw in Tom's talk yesterday, like, this has huge effects on our the outputting results with these kind of algorithms. Again, these algorithms are so sensitive to spotting the difference, and this is why your experimental or human, even just basic human variability can have a huge effect on the results. But this is why it's really important to validate your data. And this is why, as I mentioned before, these really should be used to complement and by no means replace your manual gating. Computers themselves are by no means perfect. And that is in large part, as I said, we are trying to make make computers more like humans. And this is why there's so many different approaches out there in theory to achieve the same goals, but as I'm sure we've all come across, there are differences. So we'll start a bit on clustering. So there are a range of clustering algorithms available. And as I said, in theory, the goal is very similar. They want to be able to group cells together based on how similar their phenotype is. And this is just a few listed here. As I said, I'm an immunologist. I do not fully understand how a lot of these clustering algorithms work, but the one I do have somewhat of a grasp behind is FlowSum. So FlowSum itself, the SOM within it, stands for self-organizing map. And all self-organizing maps do is essentially, we have high dimensional data here, and this is the map itself. And all it's doing is trying to place this map onto the data. And on this, each individual grid here is representative of a different cluster. And in FlowSum itself, by default, it creates a 10 by 10 grid. So that's why it gives you 100 clusters. It then goes into hierarchical clustering to create the meta clusters. But again, there are multiple different methods to go about doing the same thing. And if you're interested, Giovanna can give a 30-second k-means clustering example if you would like at the end of the talk. So when considering these kind of things, obviously we have to start thinking about which clustering algorithm may be best for you. And this is always dependent, like everything else, on your data and the question that you may want to answer. So some things to consider here, probably the most practical one is the number of cells that you want to cluster. The more cells that you have greatly increases the time it will take, and this is also dependent on the power of your computer. So for instance, here at Sydney Uni, we have access to a supercomputer that makes a big difference for us to help us cluster our data. Not everyone can do that. I think it's a pretty supercomputer, well, it's more powerful than my laptop then, at least. But yeah, that does make a big difference in terms of the practicality of which clustering algorithm may be most appropriate for your particular data set. And the other thing to consider is how many subsets you are expecting to identify. And that's because some clustering algorithms are better at identifying rarer subsets. So this is a very nice paper. Admittedly, it's a few years old, but it's still quite relevant, I think. All it's done is compared and contrasted a range of various clustering algorithms. And it's looking at the runtime of the algorithm, but also how accurate it was in comparison to manual gating. So the higher the mean F1 score, the more accurate it was compared to manual gating. So what you can see here is this is just looking at a larger population of cells. FlowSum is particularly fast, but it also retains quite high accuracy. In contrast, though, when we look at another subset of cells, so this is looking at rare populations, you can see FlowSum is still exceptionally fast, but it loses some of this accuracy. So in this case, XShift seems to be quite good at identifying your rarer cell populations. I mean, you've heard us ramble on, I think, a lot about FlowSum, and we do generally use this the most compared to the others. And I think for me personally, I use it for the speed of it. And like any sort of clustering algorithm, the assumption I have is that the clustering is not perfect. So as long as you realize that none of these, I think, are strictly bad to use, it's just that there are some that are more appropriate than others. And I'll show some examples with how I've used FlowSum a bit later on. But again, I think obviously if you have a supercomputer just lying around and you have the time, run every single one of these just for kicks, really, because there will be one that may be more or less appropriate. But I think, and we'll come to this a bit later on as well, is that sometimes it is more effort than it's worth, in the sense you could test every single one of these and it will work with that one data set you have. It does not mean it will work with every single data set that you will have. And I think, to me personally, it's more worthwhile of your time to look at the manual side rather than trying to optimize just one clustering algorithm for your data. But again, I think it is important to just be aware here that there are lots of options available. So I think another big issue that I know I have come across, and I think multiple others with these, is under or over clustering and trying to detect the correct number of clusters. And I use that term very loosely. Some of these algorithms are able to automatically detect the number of clusters. In my personal experience, I found them not to be that great. There's multiple different reasons why that could be. But I think it's important to, I guess, consider the other options are, of course, under and over clustering. So over clustering, obviously, you are going to be splitting hairs at this point. You do have more data to handle, but you can easily combine clusters. Under clustering is the exact opposite. You can get more solid subsets, but of course, you may be combining two subsets of cells together, so you can just split them. And here, I think it's important to appreciate that both of these are, can be misleading. They're not perfect. But in general, we generally aim for over clustering because it is easier to combine after clustering. But also, we just get a lot more granularity within our data set. And I guess with a lot of these things, we're hoping that the clustering or the computer will find differences that we missed. And big changes that a computer can find will be ones that humans can find as well. And this is where I think a lot of us are hoping computers can find something we may have missed. The other point I want to bring up, it's probably not the best place for this, but I couldn't find anywhere else. But I think in terms of reproducibility, and this is true for a lot of these kind of algorithms, like the dimensionality reduction ones as well, is if you, so for instance, when I used t-SNE in Flojo when it was first introduced, I realized that if I ran it, it would create a t-SNE plot. But then if I took my data into a new workspace and ran t-SNE again, it would give a different looking plot for the exact same set of data. And I'm not too sure whether Flojo has updated this, but essentially one of the things with these algorithms is the idea of it's unsupervised. And one of the draw cards of it is that you're not influencing how it starts these calculations. It's meant to be random. And again, this is one of those biases that may not be good or bad depending on your context. It's, yeah, so with Flojo, it was difficult because it just means that if it's always starting at a random point, it means even if someone has the exact same data as you, you will never be able to get identical results. And obviously in science, that is not a good thing, and especially with computers. So the way around this is we can do something where we call set the seed. So this is just the starting point for the calculation. And this is a big advantage with coding. It's very easy to do this. And it just means that if someone else, or if you run a clustering algorithm on your data set and you set a seed, you can send that data set to anyone else in the world. And as long as they have the starting seed, they can get identical results. But as I mentioned, the catch to this is that you are introducing this bias because you've removed this random nature of these algorithms, which may not be good or bad, but again, it's something I think is worthwhile to just be aware of. So yeah, clustering is pretty great, but again, it's not perfect. And like I said, going back to how computers see data differently, computers, to me at least, they're perfect. If you tell them to run one particular algorithm or to run one piece of code, it will always do that 100% of the time. The difficult thing here is that no data set is perfect, and this is especially true for biology. So this is why validation is key. And thankfully, you know, we're lucky as cytometrists because we have an excellent solution that we can use alongside this to really just validate and make us feel more confident of these results. And that is where manual gating becomes really useful. So this is just an example of some mass cytometry data that I have. So again, it's mass cytometry. That's why we've got a lot of cells banked up against the axes. This is just looking at total cells, and these are looking at two different clusters here on the same markers. So what I hope you can appreciate is when we look at metaclusters X and Y here, we can see that, yeah, there are small shifts in the level of CD38 expression, but as a biologist, I may still say these are the exact same clusters. So this to me is an example of overclustering. I'll then just simply group them together. At the same time though, in the exact same set of data that I ran with clustering, I got some underclustered data. So looking at this particular cluster, to me, these are clearly three separate populations. So you can get both under and over clustering within your own data set. And like I said before, this was run with FlowSum as well. So because these populations are so small, it could just be that it's not as accurate identifying them. But again, I ran these with the assumption it wouldn't be perfect. And I don't need it to be for my results because this is where I validate any sort of changes that I find with manual gating. And I think for a lot of these things, we're still at the stage where these kind of computational algorithms, they're useful to just give you a very quick idea of the overall spread of your data. And ideally, you will use that as an indicator for where you should focus your attention next. And that's where manual gating is still extremely useful here. So we'll move on to dimensionality reduction. There's a lot of aspects that are important for both dimensionality reduction and clustering, obviously. So there's a few more that will pop up in a bit. But one of the big things with dimensionality reduction, again, Tom mentioned this, I think, on Monday, and it's just this crowding or saturation effect. So the more cells that you try and run in dimensionality reduction, you start to get this saturation of the subsets. So you start to lose this nice, clear differentiation between some of these major cell subsets when you start to run more cells. So thankfully, there are various ways around this. And the easiest is to simply subsample your data. So one thing to keep in mind is that dimensionality reduction does take a long time. And for those in the workshop this afternoon, we'll get to run some of that and you'll get to appreciate that if you haven't already. And again, this is dependent on the cell number and your computing power. So when it comes to subsampling your data, we do this because we want to try and get a representation of all of the cells from your total data set to try and visualize them on a dimensionality reduction plot. And again, this just helps control for the oversaturation of cells. Now, there are lots of things to consider when you are subsampling your data. And a point I forgot to write down here is subsampling is affected by seed. So this is another example where what can happen is when you subsample your data, it is at random. There is always the possibility, if you have a combination of healthy and diseased patients, there is a possibility that when you subsample, it will only take the healthy patients. You may not have any diseased patients. And I think you can easily appreciate why that would be an issue when you look at your plots. So this is why it is important, again, to validate, and it can be very worthwhile to have a look at the subsample data. So some things to consider here, and again, it's dependent on your data, is whether you just do a subsample of everything. So if you have a concatenation of 100 or 1 million cells from healthy and disease, and you just cut that down to 100,000, that's one way you can go about it. Just appreciate you may not get even numbers of cells from each group. So another way is that you can make it such that each group contributes the same number of cells. When you do this or the previous one, you will also have the possibility that maybe not each individual sample will contribute the same number of cells. So you can also do it in that manner. And the other thing to consider here, through my experience at least, is that when I run, say, total PBMCs, I find I can run 70,000 cells. That's my general magic number for a quick overview. I find that quite nice. But if I'm looking at a smaller subset, and I'll show some of these later, in particular, I'm interested in B cells a lot of the time. I find that if I've just gauged on B cells, I only look at 10,000 cells. So again, there's multiple ways to go about this. And again, this is some data from Tom that he shared, and I think it's such a perfect case study for this. So essentially, we're just looking at cells within the brain of either uninfected or infected mice. And what I hope is super apparent is that there's a huge cell infiltration within the brain of infected mice compared to uninfected. So if you did a blanket subsample from here, you may not be getting many healthy cells. If you took if you made it such that each group contributed the same number of cells, again, that would influence and put more bias on the uninfected cells. Or you could have it such that each mouse contributed the same number of cells. And I think the key thing here is that, again, that there's not necessarily a wrong answer, but it really is important to just appreciate how this will impact your outputting results here. So one of the, I think, things we're scared of, obviously, is that when we subsample our data, we are throwing data away. And, you know, that there are ways around this stuff. So what we recommend and what we do is we first cluster on the total cells that we have. So if that is 1 million cells, and then from there we subsample and then run dimensionality reduction. So what we can then do is we can infer cells of the same cluster where they would reside on the dimensionality reduction plot. So this is the same t-square plot I showed before from Tom's data, and it just shows us where clusters reside on this t-square plot. So we can use this to have a look at our larger data set and do more quantitative comparisons between that larger data set without necessarily losing it for our comparisons. So something else to obviously consider are parameter choices. And this is true for both clustering and dimensionality reduction. Oh, and sorry, the other thing I forgot to mention here is that I don't have my chat open. So if you're asking questions, Tom and Giovanna will be answering them. If you're sending them to me privately, I'll have a look at them and address them at the end of the talk. But hopefully we'll be able to cover everything you're asking. So with marker selection, again, there's not really a wrong answer here. And these are probably not the answers you want to hear as well, because, I mean, yeah, like I also wish there was a simple one-size-fits-all for these kind of things, but there just isn't, because all of these things are so dependent on your own data set and what kind of question you want to answer. So in this case, I think the largest conflict, at least that I have had, and at least when I've spoken to others, is whether we include all available markers within our panel, or if we just use a subset of markers that we now can identify major cell populations. And I'll show an example or just a comparison of this. But I think the other thing that is important to appreciate is that you don't need to use the same markers for both clustering and dimensionality reduction. Again, it just depends on what kind of messages that you want to get across here. And if you do use different markers, be clear about it as well. So this is, again, some of Tom's data. Tom has a lot of data for slides and things, so he is a great source if you ever need them. In this example here, we're just looking at SCAR1 expression, again, on mock and infected mice. And this is not using SCAR1 within the Disney plots here. So again, you can kind of see some populations look like they're appearing here as SCAR1 positive. At the same time, there are some other populations that look like they are upregulating SCAR1 within the infected mice. Now, at the same time, oh, sorry, so I guess when we look at this as a whole, it generally doesn't look like there are many cells in the mock mice that express SCAR1. In contrast, though, when we include SCAR1 for this kind of plot, it suddenly looks like that there are SCAR1 positive cells appearing. But this is where, again, I guess everything is always relative. So even though this looks like there's more SCAR1 positive cells than here, the same also holds true for the infected mice. So it's not necessarily showing us different results, but there are different ways that we can interpret this. So for instance here, because it looks like there's so few cells that are positive for SCAR1, it may be that there are no other markers that were in this panel are able to identify the SCAR1 positive cells. That kind of shows us that SCAR1 is quite a unique marker in this context, where only by using SCAR1 can you start to identify some of these other smaller subsets of cells. But as I said, at the same time, we can still see some populations appearing within the mock first infected, whilst others look like there's just an upregulation of SCAR1 expression. So with this, like I said, I can't give you a blanket answer for this, but it is just dependent on what message you want to get across here, and appreciate how your choice will impact these outputting results. Another big one, especially if you're on Twitter, I guess, is whether you should use t-SNE or UMAP. And for me personally, they're both just for visualization. I don't think either one is really better than the other. This is, again, this may not be the answer people are hoping to hear, but it's just dependent on your data and what message you want to portray. So this is just an example I have. So this is a t-SNE plot, this is UMAP. This is some B cell data I have. And what you can see here, these smaller populations off to the side, they're just different isotypes of B cells. So within the UMAP, for example, it's kind of accentuating these differences. So these smaller subsets of B cells are really phenotypically different compared to the major population of B cells. The majority of these are naive B cells. So if the message you want to get across here is that these are really phenotypically unique, I think this is an excellent example. In contrast, t-SNE, on the other hand, you can see it's losing that large differentiation these subsets are, but at the same time, you're getting a lot more granularity within the naive B cells, which again make up the majority of the cells here. So in this case, if you wanted to have a look at the marker expression within this larger subset of cells, t-SNE in this example, I think would be more appropriate because we'll be able to more clearly identify potential subsets within this major population. So in our case, and with Inspector at least, we made sure there was both t-SNE and UMAP, so you can easily run them both. And this is exactly what I do for my own data sets. I run both t-SNE and UMAP because at the end of the day, I guess when I run these things, I don't think anyone knows exactly which plot they want, just without looking at what the plots actually give you. So I've kind of gone through these things. Like I said, they can be misinterpreted or misleading. And I think the biggest thing here is, again, it's just dependent on what message you want to get across. If these plots are not portraying the message that you are trying to get across, you have not made a good selection. And again, these should be complementing, again, all the other hard work that you've been doing with other sort of analyses and things. t-SNE in particular has a lot of really bizarre options. And this is where, as I mentioned before, you can spend time on trying to optimize these kind of things. There is, for instance, OptSNE available as well, which in theory can just run through a lot of these options to try and pick the most ideal one. But this is where sometimes it is more effort than it's worth. But if you are interested, this is an excellent website that just really nicely highlights some of the examples between different parameters. And in fact, Tom or Giovanna, would you be able to add this website to the chat? Thank you. Yeah, it's just such a nice way of visualizing how things like perplexity can impact the final outputting plot. So in terms of how we can kind of bring these things together, this is just a very rough example of this. So what I've done is I've done clustering, just some B cells. I then did just a blanket subsample from all of them, and then I did dimensionality reduction. So this is what the Tisney plot looks like. And in this case, we've got healthy versus diseased patients. So what I think is super apparent to start off with is it looks like this is a cell subset that is present within disease but not healthy. We can then have a look at what cluster or metacluster this subset may be belonging to. So it looks like it's about 9 or 10. So we then can refer back to the larger data set that has all of our cells and then have a look at the comparison between these cells. So in this case here, it looks like, yeah, subset 9 or cluster 9 looks like it is increasing within disease compared to healthy patients. Finally, we can refer back to the Tisney plot to give us an idea of what makes this particular subset unique. So in this case here, it looks like this subset expresses high levels of TIM1 and CD185 or CXCR5. You can then go back to manual gating to see whether any of this holds true. So I didn't do that for this example because I wanted this to be more like Schrodinger's cat style thing where right now we don't know whether this is true or not, but the result itself will impact, I think, what figures we choose to represent this. So even when looking at just the comparison within this metacluster, I think it's still up to debate with this kind of stat. But I think at the end of the day, if we find that manual gating does prove that this subset is increasing within disease versus healthy, I think these plots are excellent at portraying that because to me, I think that quite nicely shows that this is a subset that is increasing. At the same time now, if I find that this is just an artifact, this isn't real, these kind of plots are, to me, misleading. They're not that useful. But that's not to say the rest of this is not useful. You may want to still use your heat maps to visualize the heterogeneity across the subset, but you may not want to show these. And this is where, again, like to me, I think that the manual gating is really the key aspect here because that's what we're obviously all a lot more familiar with, and it really is still the gold standard here. So I think it is always important to think about what the message is that you want to portray. And as I've said, there are multiple different reasons why things like Tisney, or even the clustering, can give you different results to what we may have found manually. As I said, spotting the difference, computers are better at. The other aspect here is the parameter choices that I chose, or I set. So maybe I needed to optimize the Tisney plots a bit more, or optimize the clustering as well, use a more appropriate clustering algorithm. But even at the subsampling level, as I said, there is always the possibility that when I subsampled the data, it was just by chance, none of the healthy cells from this population were included, which is why it looks this way.
One question, do you concatenate the healthy and disease conditions from clustering, blanket subsample, and dimensional reduction?
Yeah, good question. So yes, I do concatenate all of the healthy and all of the disease. In this particular data set, I just know that the number of cells between the two groups is quite similar, so that's why I just went with a blanket subsample. But as we saw earlier with Tom's example, sometimes that may not be appropriate. So just a few take-home messages, I guess, here. So computers themselves are better at spotting the difference, but they can be too good at this. And as I've said, I think this is the crux of the issue, why we can get so many discrepancies between what humans can find and what computers can find. I think it's definitely more of a pet peeve for coding people. I think when you get clustering and dimensionality reduction mixed up, they're definitely not the same, but as we've discussed, they're both able to provide a different perspective on your data, and if you really want to make the most of them, they really should be complementing your conventional approaches like manual gating. So as we went through, clustering itself is able to group cells together based on their phenotype. This allows us for very quick quantitative data, so we can easily compare between groups. Dimensionality reduction, on the other hand, keeps the cells independent of each other, so you can very nicely visualize the heterogeneity of your data, or looking at co-expression of various markers. But in all cases, validation is really essential here, and as we've been through, manual gating is still the gold standard in that case. And again, computers do think differently to humans. And as I said, I don't think I really answered any of the questions I brought up. I think it's more important to just be aware that there isn't necessarily a bad choice, but be aware of the choices that you do make, how they will impact the outputting data. Yeah, so that was everything I wanted to go through. That was a lot quicker than I thought, but I assume there may be a lot of questions that I can finally get around to. Tom and Giovanna, were there any that you would like me to answer, or if anyone has any questions they would be brave enough to ask?
Just to save me having to type it, there's a question about having cells belonging to one cluster getting split apart on the dimensionality reduction plot. Do you want to just talk about that a bit?
Yeah, so I guess in terms of, that would be over clustering, is that?
No.
Or you mean where?
I suppose in that case, it's probably more under clustering, like a group, well, I suppose there's a couple of answers. So, yeah. Like cluster one has cells that are now on all different parts of the Tisney plot kind of thing. Like they've been split up.
Spread out across the Tisney plot. Yeah. So, and I guess that's where, again, these are like clustering and dimensionality reduction are two different approaches here. So that's why even within themselves, sometimes you will have a look at your clustering on a Tisney plot, and you'll think, why is it, like you said, cluster one may be split up throughout a Tisney plot. And that will come down to, like I said, the different approaches here, but also just the, sorry, having a mental blank for words, the subsampling of your data will impact that.
I think something that's worth keeping in mind for how these processes work is that so the clustering algorithm may group cells into groups, but the way that the dimensionality reduction algorithm sees it might be different to how the clustering algorithm does it. And one of the reasons is typically with something like Tisney or UMAP, we're only rendering it in 2 dimensions. It might see, I don't know, I've got like activated cells and non-activated cells in one cluster, but on the plot, it sees that difference more strongly than the clustering does. So I mean, as you sort of addressed, one thing is if we are not clustering with enough granularity, then yes, we might sort of be collecting groups of cells together, which really should be pulled apart. It actually doesn't, if that happens, it doesn't actually have to be a huge issue. What that can reveal is that, okay, this singular group of cells that we're trying to treat as one group of cells maybe isn't. And that's actually kind of, that's an informative thing to see that, because the visualization takes away our bias. I mean, it introduces different kinds of bias, but it takes away our assumption that, hey, this is one group. And I actually get this a lot with my progenitor groups, because I'll, gate, I don't know, myeloid progenitors or something, and on Tisney, they'll be split into their different lineage islands. And I'm like, well, actually, there doesn't have to be a right answer there, because they're all connected. But it just, I suppose it suggests to me to think, to change the way I think about how I identify those populations, because it isn't exactly plus or minus. that's not exactly a firm answer to that question. I think the idea is that it's informative when that happens, and it maybe encourages us to reevaluate how we're grouping cells together. I think when you don't get that kind of cluster fragmentation, what it kind of indicates is like you're identifying subsets that are consistent and that are, I suppose, in terms of the data structure, are consistent internally, and that's probably quite a good thing. So like blood, that would probably be quite easy most of the time. Like a tissue digest with like weird infiltrating cells, that's probably when you'd get more of that kind of an effect. Hopefully that was the actual question that you asked, and we didn't just misinterpret it. That's always possible.
So another question earlier just about the PCR gamma delta cells, which are very low in number. So I think this is, you know, as I mentioned before, with the clustering algorithms, I'd suggest giving X-shift a try, just because that one appears to be better at getting those rare populations. But that's an example of, you know, a lot of these things to a computer, a lot of them are kind of density based. So they're looking at where the major populations, because as we discussed, the computers see things differently. So in terms of similarities and things, this is where they can very easily kind of ignore those smaller cell subsets, because to a computer, based on the algorithm, it may just see them as artifacts, because there's obviously always going to be some background. But I think that's just an issue that at the moment, I don't think there's a solid answer for it.
Alternatively, if the interest is in finding rare cell population, what we often do is purposely get the algorithm to overcluster. So for instance, if you're expecting probably like, I don't know, 8 populations in general, but one of the populations is very, very small, what I would do is maybe get something like FlowSum to generate, say, 16 clusters. So what that inherently does is just allow it to sort of divide the major population into smaller group, but at the same token also allows it to find the smaller subset, if that makes sense.
The other thing to keep in mind is there is a, to some extent, I mean, you know, when we do like gating, for instance, there is this kind of effect where we essentially just negate frequencies. Like, so I know T-regs are very infrequent, but we sort of ignore the frequency of different subsets and we just look at the profile to get down to them, which actually helps, because when we have clustering, it basically makes decisions for us based on the structure of the data. One way of getting around that is if you're like, all right, I've got some rare subsets. So in my in my work, I'm looking at bone marrow stem cells in amongst all the other cells, and you know, I don't get a clean separation. Well, okay, often I do, but sometimes I don't get a clean separation of like long-term stem cells versus the intermediate progenitors, things like that. If I'm clustering whole bone marrow, what I can do is say, okay, I'm going to select everything that is c-KIT positive, or you know, in the stem cell cluster group, take that out and do a whole another analysis just on the stem cells. And then I get my full resolution of long-term progenitors, short-term, you know, the intermediates, everything like that. And so you can kind of treat it like a multi-level analysis. Because it's exploratory, you can like dive into arbitrary level of, and with the system that we have, if you cluster using something like FlowSum, which is very fast and scales well to large data sets, you can then extract clustered cells from that data set, run t-SNE, run clustering again, and you can go to any arbitrary level of depth in the data set. The only limit is the cells that are actually in there. So if I have, say, you know, 1,000 stem cells in my whole data set, I have 1,000 stem cells that I can pull out, analyze in isolation, and then if I want, you know, label them, integrate them back into the full data set if I really want to. So there's a whole slew of ways that this can be tackled depending on what kind of the outcome is. A lot of my work is multi-level analysis. Like I'll split my lineages up, have a look at them in more detail separately, and then stitch them back together. That works quite well, but it depends on exactly what the objective is.
There's another question from Xavier, just following on from Shirley's, about the healthy versus disease and subsampling, either individual samples before running these algorithms. That is always an option. And I think, like I mentioned, there's not necessarily a wrong way to go about it. That is a nice way because you're starting to control for the inevitable variability between individuals. But obviously the downside maybe is that you are losing cells. And I think that's probably the biggest reason why I just subsample from a whole population as well, and I do the clustering on every single cell I can get my hands on. But it's a good point as well, I guess, especially with like everything I presented here. I think the biggest assumption it makes is that your data are ready to go and there aren't going to be any sort of batch effect issues or alignment issues. And for a lot of the time, obviously, that's not the case. And I think that's where another option, if you have enough batches, so say if you have 10 batches and it was only one batch of experiments that look awful compared to the rest, just don't run these algorithms on that one batch. You can run it on everything else because at the end of the day, like I said, we do recommend going back to the manual gating. And this is where, again, humans are better at pattern recognition, so we can just subconsciously control for any sort of batch effect because we have an understanding of obviously the data, but also just the biology behind these things.
Probably another thing worth pointing out, just like any other sort of data mining, machine learning, or those fancy software, generally speaking, all this clustering algorithm, the more data you give it, and this is also the same for dimensionality reduction, the more data you get, the more informative, the more information you give it to sort of make a decision and stuff. So if you want to subsample, I would recommend subsample just for the visualization. We do not subsample before you do clustering.
There is kind of an interesting, actually, discussion point with that. So if, I don't know, if one of your samples had, let's say, 10 million cells and one had 1 million cells, then the 10 million cell sample will have more influence over the data structure than the other one. In some situations, this is actually kind of appropriate. Like if with the brain infiltration data, there is more structure provided by the samples that have cellular infiltration into the brain. And so it actually kind of is appropriate that way. If you're dealing with something like PBMCs, then, I mean, so there's kind of this idea of like, okay, well, maybe you'd want to say, I'm going to just start with 1 million cells from each sample so that it's all nice and even across the board. And I think that thought is correct, rather than having 10 million from one sample and 1 million from another. However, the reality of what happens there is you say, okay, I'm going to go for equal numbers of cells from each sample. There's always going to be one sample that has like 1,000 cells. And you're like, well, do I just do 1 million from everything and then 1,000 from that sample? Or, to an extent, particularly at the clustering level, do you just say, look, I'm going to take whatever I've got, we're going to cluster it, and then make some decisions about how it differentiates after the fact. And I think it's one of those kind of just the real world, grit of doing that is we often end up just being like, okay, we're going to throw whatever cells we have in there. If there is a situation where I've got, I don't know, 19 PBMC samples with 1 to 2 million cells in each, and then there's one with 20 million, okay, in that situation, I don't really know how I'd pull that off, but in that situation, okay, I'd probably treat things a little bit differently. Otherwise, if they're all in the same kind of ballpark, I think just the real world version of doing this is I'd probably pull that all in for clustering. So I've got as many cells as I possibly can. And then at the point where we're doing comparisons, we start subsampling for visualization and doing that in a sample specific way, which I think is important, particularly if you're dealing with tissues and things like that. For blood, you can usually just go sort of equivalent cells per sample, depending on exactly how you're tackling things.
Next thing. Is it possible to run Sydney or UMass just on individual cluster? If you want to drill down into the cluster, you can just do that.
Yeah, do it all the time.
You don't have the overcrowding problem.
Yeah. It's not online yet, but we have a multi-level analysis workflow that essentially does that. Oh. Just pull out the T-cell cluster and analyze it. Yeah. Not online yet, but it will be shortly. Actually, just for interest, there's an algorithm called HSNI, which is an adaptation of t-SNE, which does something similar. It kind of, in effect, runs t-SNE on
clusters of cells, and then within each cluster of cells, you kind of get a new Disney plot almost. That probably doesn't make a lot of sense just saying it in words, but it's a hierarchical sort of set of dimensionality reduction coordinates, which I think is quite a cool way to address it.
The other question I can answer there. So I have personal experience over this. So I've run an experiment whereby I first run clustering and then the measure the reduction for visualization, and or I do the measure the reduction first and or then run clustering. The answer is I would strongly recommend option 1. Based on my experience, when you run option 2, you lose a lot of the granularity of the each individual cells and the nuances in the phenotypes. Because, I mean, what the measure of the reduction originally in ComSci made for is for the purpose of compression. If you want to compress a massive data into a smaller representation, while at the same time maintaining the differences between different groups. So yes, I would definitely stick with 1 rather than 2.
There's actually an important thing to bring up here. There's often this kind of question of like, can I just cluster on my, say, Disney parameters? So, you know, just clustering on Disney 1 and Disney 2. It is tempting because there is this situation where you're like, well, there's an island that's, you know, an island of cells that's present in one sample but gone in the other. And so can I just cluster on the Disney parameters to get that as a group? The answer is that you shouldn't, as a general rule. So, I mean, okay, I can see exceptions where maybe you, like, it does kind of very much depend on the objective, right? But I think as a general rule that shouldn't be done, because dimensionality reduction is awesome because you can take this complex high dimensional data and plot it in 2 dimensions in a meaningful way. And that's just super, super helpful. What it does though, is it does remove a lot of information about how different those cells are. So when you cluster on dimensionality reduction, for instance, what you might find is that, let's say there's 2 kind of Disney islands, let's say, that are near each other, and maybe they get linked together in a cluster. Now, the actual absolute position of those islands is not meaningful. And so they might get linked together for reasons that have nothing to do with their similarity. And so you've kind of created a pretend cluster, as it were. And so I think, so clustering should be done on the kind of, on the data itself, not on the derived parameters like Disney and things like that. And so in this situation, if you were just running, say, both clustering and Disney on the cellular markers themselves, in theory, you can do it in either order because they don't affect each other. Like you know, do clustering first or do dimensionality reduction first. As long as it's all being run on the same markers, then it doesn't, you know, matter in theory. What does matter is the fact that dimensionality reduction will hit a limit in terms of the number of cells that you can run. So if you do clustering first, where you do have scalability, you can do it on 30 million cells, then you could subsample and run some Disney or whatever on the subsample data and then get a meaningful plot. But as long as all of that is being performed on the cellular parameters themselves, then, you know, if cell numbers aren't an issue, then the order doesn't strictly matter. But we find it more practical because, I mean, no one's really running experiments with only 100,000 cells unless you're in single cell sequencing or you're dealing with like really rare subsets. Most of our data sets are, you know, at least 10 million or more. And so that approach makes more sense to cluster large, subsample it, run Disney on the clustered data, and then interpret the results that way.
You still lose stuff though if you run Disney first. I think you still lose information if you run the measurement reduction first before you run clustering, even though they're both using the same subset of markers, which is stable markers. Because how I see it is that if it is an island in Disney, it doesn't necessarily mean it's a separate cluster, because all it does is just accentuate some differences that it can find. But what I'm trying to say is that an island in Disney or UMAP doesn't mean that, you know, it's a cluster. The grouping that is generated by clustering is more nuanced than the grouping generated by something like Disney. That's what I'm saying.
Well, yeah, I think as well, like it's, again, what you are sacrificing, the whole point of computers is that you can look at more than 2 dimensions at once.
That's right.
And as soon as you're cutting or doing dimensionality reduction, as the name suggests, cutting high-dimensional data into 2 dimensions, you're just like really sacrificing the huge advantage that computers have in the first place. Good to hear we're all on the same page. Cool. Are there any more questions, or does anyone want to, or brave enough to ask? You're more than welcome to.
Don't be shy, no one's gonna bite you.
Actually, there was just an interesting point I wanted to make. If people visit that link that we posted, that distill whatever, which kind of explains how Disney works.
Did you want to share your screen?
You know what? That sounds like a better plan. I need to go find the example that I was making. I think there's one of the things that we need to keep in mind with how we use these tools is local versus global structure. And we have talked about this, so I'm repeating myself a little bit, but it's worth keeping in mind that at any point that you run, say, something like Disney, in its default settings, we compromise the global structure of the data to be able to prioritize the local structure. What this means is that broadly, cells that appear within islands tend to be, quite related to each other, but the position of the islands sort of objectively aren't meaningful. So if you had two islands near each other, that doesn't mean that those islands are similar, but the cells within each island are. There's this great example of how dimensionality reduction works online. There's a Reddit thread about it, actually. Oh, maybe I won't be able to find it. Okay, we'll post it later for a retrospective. But just having a look at how this website works, there's a couple of great examples where you can see how data is influenced when you run Disney. So you can select a starting data set, so maybe a grid of points or like a circle or something, and you can kind of see how Disney will deal with it depending on the settings that you set. Now in this case, at some point this should get all warped. Oh, maybe this one's fine. One of the key parameters that influences Disney is this thing called perplexity. Now perplexity essentially says whether we're trying to look for local relationships or global relationships. Local essentially is saying that I want to look at really fine-grained collections of cells at the expense of the overall data structure. High perplexity says I don't really care about the fine-grained stuff. I just want to see how all the points relate to each other. So this is a real life experiment, but if I turn this down, then we shouldn't get it. Oh, damn, we did get a grid. All right. In theory, what I was hoping would happen, I know it'll max out if we do it that way. Let's try with a couple of these somewhere in the middle. So this is Disney run on two groups of cells. And if we have a low perplexity, what we hopefully will see here is it will start creating some random groupings, right? And so now this is what the data looks like to start with, a group of yellow cells and a group of blue cells, but Disney's kind of stretched it out in kind of all dimensions here. Now this doesn't really represent anything meaningful. It's essentially collecting noise and interpreting that as a difference. Now, as far as the computer is concerned, it's accurate, right? Like it doesn't know it's noise, it's just showing difference. We, I guess, know it's noise because we can interpret that. And biologically speaking, this is the same thing. So I can ask Disney to pull this apart in more and more detail. If these were cells, I could go, are these meaningful differences or is this just noise in the data? If I turn perplexity up, let's just dial it up to something silly, see if it actually manages to render it properly. Oh, there we go. It's produced almost the same plot because we've asked it to, you know what, don't look for fine-grained relationships. We don't care about the nitty-gritty. What we want to see is the global structure of this data. And in this case, the global structure is basically two completely different groupings of cells. So that's a really nice example where depending on what we input here, we get different results. But that kind of highlights the fact that this is a data-driven process, and the interpretation of that needs to relate to something biological. And there's this great kind of, story in different contexts where it's like, well, I found a novel cluster, and that's fine. A novel cluster is something to be excited about, but when does a cluster become a population? And to do that, we essentially need to, ideally, find a way to sort those cells, take them out of a system, put them in culture, see what they do other kinds of studies to figure out, is this biologically something different to what it was before? Otherwise, it's just a feature of the data. And that's an important, perspective to add to anything which we approach from a data-driven side of things. So it doesn't make it, wrong to have a novel cluster, but it makes it incomplete unless it kind of goes the full scientific journey, so to speak. And that's, I think, just an important thing to keep in mind with how we address these things. Oh, yeah, actually, I'll just deal with Laura's question first. Sorry. Tisney's default is a perplexity of 30, which tends to work for most cytometry data sets. There's a whole storyline behind why that is, but it's essentially the performance has got a fairly shallow curve there. And for the type of data we have with the number of features we often have in cytometry data set, a perplexity of 30 just happens to be suitable most of the time. You will find though, if you run it on, let's say you ran Tisney on 4 or 5 cellular markers, then a perplexity of 30 will go to too much granularity and you'll get these groups of cells splitting off randomly. That's just driven by noise effectively. So you do see some factors like that. One of the best options is to run multiple iterations and see how that matches what you understand of that cellular system. That's probably one of the best things to go. But I mean, just start at 30. There's not a good reason to start with something different. That's probably a general rule for most things. Just start with the default because you've got to start somewhere. But you can run a couple of them with different settings and see how it looks. Oh yeah, so there's a good question here about which clustering method do you pick for a discovery type approach. So FlowSum requires, it doesn't actually require you to necessarily specify the clusters. You have to specify the grid size, so like the level 1 clusters. You don't have to specify meta clusters, which is the kind of the groupings of the clusters. You can let FlowSum decide that for you. So it'll tell you, okay, I've found 15 groups of cells that I think are meaningful. We don't typically like doing that because I guess I'm the one who ends up deciding if it's a meaningful population. So I want as many clusters as possible. I can then decide, okay, these 2 clusters are the same population. I guess because I like having that sort of control. And even if it's something novel, I can go and make a decision about that. So it's fine to let FlowSum decide. I think best practice is you decide, aim high, 40 metaclusters. I don't know, it depends on what you're analyzing, but aim high and then make decisions about that output, I think is the sensible way to go.
Well, maybe you can just get it to sort of determine the number of metaclusters first. Look at it in the UMAP or the Tisney plot, see if it makes sense, and see if you want to add more clusters or reduce it. It's just a starting point.
Yeah, that's a good idea, actually. Let it give you an idea of what you might be doing.
Yeah.
I think the other thing that I've come across as well with that is, like I said in the example data I had, where you get both over and under clustering within the same data set, which is obviously less than ideal. So that's why sometimes if you think, it's identified 6 of the 7 clusters I want, I'll increase it by 1. It doesn't always work that way. And I think that just comes down to, again, how the algorithms are able to kind of comprehend your data. And a lot of the time they may see that the differences between one subset may be larger than identifying a new, smaller subset of cells. And I think, yeah, like Joanna and Tom suggested, you can experiment with those numbers, but it does hit a stage that I know I've spent far too long with some data sets trying to find the perfect number, but sometimes I don't think it's possible.
There's a great question here about comparing like experiences with phenograph. So that's another clustering algorithm. It's a graph-based clustering algorithm, which is different to how FlowSum does things. I have two comments. One is that in my experience of phenograph, when it was originally implemented, FlowSum was much faster. And I mean, most of my decisions came down to that, really, because I could get my clustering done in 10 minutes rather than, I don't know, an hour, whatever it was. There have been changes to phenograph recently, which have improved the speed enormously, but you probably have to make sure you go find the right version, which has been sped up. I think the key difference in terms of performance is phenograph will, in my experience, create more arbitrary divisions of things in ways that are maybe not quite so helpful. This isn't an absolute rule though. This isn't kind of in every case. So if I take this example here, this bone marrow data set, this big cluster here is my red blood cells. And essentially all of them in the data set are phenotypically, let's say, identical, or as close as they can be to identical. I know that FlowSum will put this in one big group because there's nothing to split apart. Phenograph in a default usage will split this into a bunch of groups that are effectively arbitrary. Now, FlowSum would do the same if I asked it to turn up the meta clusters or whatever, but I think what FlowSum does here is it sees the structure of this as more consistent, whereas phenograph will try and find deviations in it. Now, maybe that's actually good. Maybe I need phenograph to give me a bit more kind of, I suppose, division of the data, but overall, I preferred the way FlowSum does things, but I think it's one of those, it's context specific. That's the way that I've used it on my data sets. So it's not really an absolute statement. I really like phenograph. I used that consistently before I used FlowSum. And particularly now that phenograph is faster, I plan to re-implement it and just, I don't know, in an analysis, right, run both, see where you get consensus between the two. There is no right answer. There's just different ways of dealing with it. So, I mean, I don't know, run 10 different clustering options and see where you get consensus. And I think that's, well, probably the smartest idea. A few new approaches from a few groups essentially do that. They run different iterations of one algorithm and sort of multiple algorithms simultaneously. And then the final quote unquote clusters come from consensus between them. And then you get sort of a score for like what clusters are, you know, in every case are being separated and what things are more questionable. And I think that approach is probably the better practice here when you think about it.